Hello, Mariam.
Hi, doctor.
Can you tell me where we stopped?
In respiration paper 2?
Yes.
Yes, I'll check now. Page 8.
Okay. So we stopped at that question number 5.
Yes, we did it so.
Wait for 2 minutes exactly. Can you see my screen now?
Yes.
Okay, hopefully we will try to finish all the 32 pages. I hope. Okay. This question here, let's read it. When tobacco smoke is inhaled, chemicals such as nicotine, carbon monoxide enter the circulatory system through the gas exchange system. Tar will build up on the lining of the gas exchange. Many people decide to give up smoking tobacco in order to improve their health. Some of the structures in the human gas exchange system through which tobacco smoke passes are shown. Okay, now describe the gross structure of the human gas exchange system. This is a very easy question. The first thing you will do, you will look at the number of marks, 3 marks. So you will be generous in your answer. So look at the diagram and how will you describe the gross structure? This is more than easy.
What does the gross structure mean?
The total structure, the whole thing. What do you see? Like, what are these structures that you see?
The trachea, it contains the C-shaped cartilage.
Yes.
The also it then divides into two bronchi,
Yeah.
which also contains cartilage.
Yes. What's the difference between the cartilage and the bronchi and the trachea?
And the trachea, it's incomplete C-shaped rings.
Very
And
good.
the bronchi, they are just in different places. They're not connected.
Yes, they are separate plates or hoops. Hello, Kanzi.
Hi,
Okay,
doctor.
and then at the bronchi divide into smaller
Bronchioles.
bronchioles. What is not present in the bronchioles?
The cartilage.
And no cartilage and no,
Goblet cells.
yes, even though you don't see that, but it's okay to mention it. Now, at the end of the bronchioles, Kanzi, what do we have?
The cartilage.
At the end of the small bronchioles, these small sacs.
The alveoli.
Yes. Okay, write with me, okay? Don't waste time. Write with me as we're writing. At least write points. Alveoli, you can see, okay, now we want to describe what you see. What can you say? Like, this is out of 3 marks. What do you see also from the picture? Because they are only asking you to describe like the anatomy. Think of something to write. We want to write more.
There is a lot of alveoli in one lung.
Yes, you can say many alveoli. to increase the surface area, for example. You can compare between the trachea and the bronchi in terms of diameter, because this is something visible. The trachea is wider, bronchi narrower, bronchioles, they are the narrowest. If you look at the mark scheme, let me look at the mark scheme. I think they remembered we solved this last year, and they mentioned something about the nose and the mouth, even though it is not obvious in the picture. It's not in the picture at all, but it's in the mark scheme. If you see something like this, I think nose, mouth, nostrils, nasal passages, I was surprised when I saw that. But this does not mean, when you see it in the mark scheme, this does not mean that it has to be present in the answer. Any three from, okay? So we mentioned the trachea, bronchus, bronchioles, alveoli, lungs. We see the lungs, so we should write lungs. And then Maryam mentioned that it divides into two bronchi, and we mentioned the sequence, I guess. And then we said that the trachea has C-shaped incomplete rings of cartilage, while the bronchus has plates, even though this is not visible, but we mentioned that. This is an important point in my opinion. And we mentioned the diameters. Okay, so we mentioned more than 3 points. So what we mentioned will give us the 3 out of the 3. Okay, you will realize that the questions are actually relatively very easy. Soon after a person stops smoking, the short-term effects of nicotine are reversed. State changes that will occur in the cardiovascular system as a result of reduced nicotine levels. So you will remember what the nicotine does and what is the improvement when you stop the nicotine. What does the nicotine do? Yes, Farouk.
It increases heart rate and blood pressure.
Yes, you are very correct. The nicotine increases heart rate and blood pressure. Why? Because what is the first word that you will remember when you discuss nicotine?
It's addictive.
Okay, it's addictive. This is correct. But what you said is not because it is addictive. Like, let's write what the nicotine does. One, it's addictive. I know you did not study, so try to recall what you remember from class. It's addictive. What else does the nicotine do? Before we start, remind me, the nicotine goes to the blood or remains in the lungs?
It goes to the blood.
Very good. So the nicotine will go to the blood. And like Farouk said, yes, it is addictive. What else, Mariam? You wanted to say something.
They cause vasodilation.
You're sure of what you're saying? Vasodilation?
Sorry, constriction.
Yes, vasoconstriction. Perfect. And from there, the vasoconstriction means that the arteries or the vessels will become narrow, reduce in the blood flow. And because they become narrow, the heart will have to pump stronger to compensate for this increased resistance. So due to the vasoconstriction, this will result in what you said it, Farouk, once we started.
The increase in blood pressure and heart rate.
Yes, the increase in heart rate and blood pressure. This is correct. And if you write it, you will get a mark for that. But I want you to understand, why does the heart rate increase? The heart rate increases for so many different reasons. When you're scared, when there's adrenaline in your blood, when there's cortisone in your blood, the stress hormone, when you have, what's this called? When you have problems in breathing, when you have increase in blood pressure, etc., etc., etc. So the heart rate and the blood pressure increases, yes, but we want to know why. Because the nicotine does something very, very, very conspicuous. This is special for nicotine. It causes vasoconstriction. Now, when you are asked about the carbon monoxide, what will you remember? I know that's not in the question, but let's revise. Carbon dioxide causes what? It binds with what?
Hemoglobin.
Yes, it binds with hemoglobin instead of the oxygen to give you carboxyhemoglobin. This reduces blood flow, and eventually also it will cause an increase in heart rate and blood pressure. But the difference is how we reached there. The nicotine causes vasoconstriction, and the carbon monoxide causes a decrease in the oxygen levels. In the end, you will have the same signs and symptoms: increased heart rate, blood pressure, difficulty breathing, inflammation, chronic bronchitis, etc., etc. Okay, I don't want to dive deeper. Let's answer the question. We said nicotine will cause addiction, vasoconstriction, like Farouk said, increase in heart rate, increase in blood pressure. What else does it cause? How does it affect your platelets?
They become sticky.
Very good. It makes the platelets sticky. Okay. How does it affect the cardiovascular system specifically? What does it do to the inner walls of the arteries? I'm trying to remind you. Tell me, Kenzie. What does it damage?
The arteries.
Yes, it damages the artery. Which layer of the artery? The innermost, the middle layer, or the Yes, it damages the endothelium. Don't forget. And when it damages the endothelium, it will help in the buildup of plaque and blood clots, and it can cause coronary heart disease or heart failure. Okay, so you will remember, you will see the word nicotine in the question. You will remember all the bad things that the nicotine will do, and then you will answer the question. State the changes that will occur in the cardiovascular system as a result of reduced. This is someone that was a smoker and then he stopped smoking. So he will not be addictive anymore. He will have vasodilatation. His blood vessels will not constrict. He will have the opposite of vasoconstriction. His platelets will not become sticky. He will not have high chances of blood clot formation. These chances will become reduced. He will have a decrease in heart rate, a decrease in blood pressure, the opposite. Why? Because he stopped. And he will have less chances of developing coronary heart disease. Okay, and of course he will have better oxygenation and he will have more energy because of the better oxygenation, etc., etc. This is a 2-mark question, but that same question can be out of 3, can be out of 4. That is why we're mentioning everything, so that we can remember everything we have to write in case it's out of 4 marks. Let's look at the answers if you want. Okay, blood pressure decreases, platelets are no longer sticky, the endothelial repair, endothelium is no longer damaged. Okay, I think we did a good job. Any valid point? Any valid point means increased in the peripheral circulation, which is increase in blood pressure. Blood pressure, when it rises, it causes an increase in the pulmonary pressure and the systemic pressure. But we said that the pulmonary pressure causes the most damage. Okay, blood vessels diameter will increase, no vasoconstriction, we will have vasodilatation. I think we mentioned everything. Any questions? Easy, yes? Okay. Now answer with me while we are writing answer. Figure 5.2 shows oxygen dissociation curves. Curve A here shows measurements obtained from a person who is a heavy smoker. So A is a smoker, heavy smoker also. And B shows measurements obtained several weeks after that same person stopped smoking. So B, he stopped. Okay, very nice. Kenzie, what happened to the oxygen dissociation curve? It was at A and then it moved up to B. So how will this affect the affinity of hemoglobin or the percentage saturation of hemoglobin?
It will decrease.
Kenzie, how come will it decrease, Habibti? A, he's a smoker. He's a heavy smoker. He stopped smoking. And the oxygen
It
dissociation
will increase.
Yes, this is a big difference, my dear. Be careful. So the oxygen dissociation curve will go up. This means that the affinity of the hemoglobin will increase. The hemoglobin now is happier. It will bind with more oxygen to give me oxyhemoglobin. Percentage saturation of hemoglobin will rise. Now let's see, what do they want? Okay, with reference to figure 5.2, describe and explain how the results show some of the health benefits of stopping smoking. 4 marks. So here, this question wants you to look at the graph and get, of course, data from the graph, and they said it clearly with reference to figure 5.2. Most of you will throw the information you know about the health benefits of stopping smoking. You will say a decrease in nicotine, a decrease in carbon monoxide, a decrease in tar, and all the bad effects will go away. And this is correct, but unfortunately, this is part of the answer. And you forget to refer to the graph. So when they tell you describe the results, or when they tell you refer to the figure, then please refer to the figure. They said describe and explain. So this question that is out of 4 marks should make you worry. There is a describe part and there is an explain part. Okay, now, Mariam, how will you describe?
As
We
the
actually
person,
answered the question. Excuse me?
as the person stopped smoking,
Yes.
the affinity of hemoglobin to oxygen increased.
Very good. So the affinity will increase. Yes. What else will you say? You will do what? You will refer to figure 5.2. How do you refer to figure 5.2?
The oxygen dissociation curve increased, and
Okay.
the highest percentage saturation of hemoglobin increased from 76 to 96.
Okay, this is what I was waiting for. Very good. She used numbers from the graph. I was waiting for that. She said that the affinity will increase and the oxygen dissociation curve will move up. Yes, it will move up. Very good. But still, this is not what they want. They want numbers, any numbers. Give them any numbers. Maryam said what? Maryam said that the maximum will be 76. And this is when he stopped smoking. As he's a smoker, the value of A is around, what were you saying, Maryam? Maryam, this 76 referred to what?
When he was smoking.
Okay, 76. And then the affinity increased to nearly 90 something when he stopped smoking. So she used numbers. This is correct. Something else, I'll give you an advice. Something else is that you can use the numbers for the same partial pressure of oxygen. For example, when the partial pressure of oxygen was 8, when he is a smoker, the percentage saturation of hemoglobin was here. When he stopped smoking, when the partial pressure of oxygen is still at 8, when he stopped smoking, then the percentage saturation increased. So this shows that at the same partial pressure of oxygen, the affinity increases. Okay, so you will support your theory. Your theory is the affinity increased. Now prove it using the graph. I'm going to choose one particular partial pressure of oxygen and show that when he was a smoker, the percentage saturation was less, and when he was a non-smoker, the percentage saturation was higher. This is just an advice because this is how we do it in biology books. But what Maryam said is also correct. Okay, so this is the describe part. Now explain. Farouk, can you explain why will the affinity increase and why will the percentage saturation be better and why will the curve go up? Can you explain?
Because of the board shift.
Yes, we did say that. This is the describe part. In the describe, Maryam said it all. She said that the affinity will increase, and she said that the graph will move up, which is the Bohr shift. And Maryam gave us numbers. She used numbers from the graph. So we fulfilled the describe part. Now explain, why does the affinity increase? Now, again, when you are talking about affinity and hemoglobin, this reminds you of which chemical in the cigarette? We have nicotine. We studied 3. We studied nicotine, we studied carbon monoxide, and we studied tar. And there are
Carbon
other
monoxide.
chemicals. Exactly. The moment you are referring to the hemoglobin, then directly point at carbon monoxide. So can you explain now using carbon monoxide in your explanation?
The carbon monoxide, there's no carbon monoxide, so the hemoglobin will have more affinity to the oxygen, so it will bind to the oxygen instead
Who
of
will
the
bind to
carbon.
oxygen, the hemoglobin or the carbon monoxide? I did not hear you well.
The hemoglobin.
Of course. Now, when there is less carbon monoxide because he stopped smoking, the hemoglobin will be able to bind with what? Just like you said, the oxygen instead of the carbon monoxide. You can explain further that when the carbon monoxide was there, the hemoglobin has a higher affinity. I heard you saying this, which is very good. So the hemoglobin will carry more oxygen to give you oxyhemoglobin, and this will be delivered to the tissues to use it in aerobic respiration. Okay, and this explains, hopefully, this explains the change in the shift. You actually can say a lot of things because we studied this graph in detail. You can say that the oxygen concentration is higher at the lungs or at the respiring tissues. Where is the oxygen more present? Come on, the graph, we have a graph here. Percentage saturation of oxygen and the affinity. Now we broke down this graph in detail. We said the graph looks like an S and we broke it down in detail. I expect you to know it. You just had an exam. Here, when we have lots of Oxygen. And the affinity for hemoglobin to oxygen is high. We have lots of oxygen where? In the lungs or in the respiring tissues, Farouk?
In the lungs.
Of course in the lungs. Of course in the lungs, okay? And if you want to write further, you're worried and you want to write further, you can describe the shape of the graph. Here, there is a slow pickup for oxygen because the affinity is low. And after it picks up the first oxygen, there are conformational changes, remember? And then the hemoglobin will pick up the remaining oxygen very, very, very quickly. That's why that part of the graph is described as being very steep. A small increase in the partial pressure of oxygen results in a great increase in affinity. And then up there, the affinity is high, but the picking up of oxygen is slow because the hemoglobin is saturated. Didn't we take all this? Didn't we?
Yeah, with this.
Okay, okay. Now let's look at the answers. I'm sure, like, I don't want to waste time, but okay. Assume from point of view in lungs so that at high partial pressure, so more oxygen taken up by the hemoglobin. Okay, now we understand that. Before and after data. The most important thing is the data. Please, the data. They want data. Calculated difference in data to support. This we did. Okay, carbon monoxide is no longer binding. This is what Farouk said. There is less carbon monoxide and the hemoglobin will bind with the oxygen. There will be no carboxyhemoglobin no longer forming. We said it. Hemoglobin has a higher affinity for carbon monoxide. We said it. And also we can mention that they have the same binding site. We took it, but we forgot to mention it. See? Okay, 5, more oxygen dissociates from oxyhemoglobin at low partial pressures. Yes, this is what I was telling you. Describe the graph. At low partial pressures, there is less affinity because the hemoglobin will bind with the what? Hydrogen ions instead of the oxygen. You don't have to write all the 8 points, but we will get the full mark. Hemoglobin is less saturated with oxygen, except hemoglobin has less or decreased affinity for oxygen at low partial pressures. Before and after data calculated, we did that. Reference to increased allostatic effect or regains ability for conformational change. I reminded you to mention that. Okay, yes, we will deliver the tissues to the oxygen. And here are the consequences of less or decreased oxygen. Now, can you remind me what are the consequences for decreased oxygen in the body in general? When I have less oxygen.
More anaerobic respiration.
Very good. More anaerobic respiration. What else?
Increase the concentration of lactic acid in muscles.
Very good. More lactic acid and the blood is acidic. And what about your energy levels? High or low energy?
Low energy levels.
Low energy levels. Also, how will this affect your breathing rate and heart rate?
It will increase.
Yes, to compensate for this lack of oxygen. And this is going to affect the pressure in the lungs, causing pulmonary hypertension and strain on the heart. So you have a lot of things to say, but okay, any questions? Nice and easy?
Yes.
Okay, Farouk, this question is for you. A person who gives up smoking decreases their risk of developing lung cancer, a non-infectious disease. Explain why lung cancer is described as a non-infectious disease. We will take the type of diseases in the next lesson, immunity, but you know now what is a non-infectious disease. We took it last year. What's a non-infectious disease? Say two things.
A disease that can't be transmitted from an infected person to an uninfected person.
Good. This is one point. Non-transmissible. Okay. Is it caused by a pathogen or not caused by a pathogen?
Not caused by a
Yes, not caused by a pathogen. Okay, write this down. I'm writing it with you. Write it down. We want to solve together. Not caused by a pathogen. These are the 2 marks. So mentioning these 2 marks, this is enough. And explain, elaborate. Not transmissible means it will not be spread from one person to another. Not caused by a pathogen, give examples. Okay, when you see the word explain, just be generous. Not caused by a bacteria, not caused by a virus, etc., etc. And
So,
also,
can I say
the
that
word,
it's mutation?
huh? What do you say? What did
Can
you
I
say?
say that it's like it comes from mutation?
Of course, I was just going to explain to say that. Of course, it's cancer. The moment you see the word cancer, right, it is caused by mutation, proto-oncogene to oncogene. It will inhibit the tumor suppressor gene, but it's only 2 marks, so this is very, very enough. Okay. Oxygen enters the bloodstream from the alveoli in the lungs, and carbon dioxide leaves the bloodstream to enter the alveoli. Most of the oxygen is carried by hemoglobin in red blood cells to the body tissues. Now outline how oxygen enters the bloodstream from an alveolus. This is such an easy question. So you know what do this question want? They want to describe the direction of movement and the method of movement of the gases. We have a nice alveoli here, and we have blood capillaries. Okay. And where is the oxygen? Where is the CO2? How does it move? In what direction? What does it cross? This is what they want. So who will answer this easy question? Wake up with me. Hello? Where's everyone? Can you hear me?
Yes, we can hear you.
Kenzie, can you tell me the direction of the oxygen movement? Oxygen will move from where to where?
From the alveoli
Very good. So the oxygen will move from the alveoli down to the
to the cells.
to the capillaries, to the surrounding capillaries. Okay. How? What is the method of movement? This is so easy.
By
This is an
diffusion.
O-level question. Of course. So the direction
Diffusion.
of movement, oxygen, yes, write down with me. It will move from the alveoli to the surrounding capillaries. Of course, by diffusion. Of course, from high to low concentration or down a concentration gradient. Now the carbon dioxide. Farouk, can you tell me the direction of movement? It will move from where to where?
From the capillaries to the alveoli.
Yes. Like, of course. So from the capillaries, write with me. I'm writing with you word by word. Into the alveoli, again by diffusion down a concentration gradient. Okay, this is 3 marks, so I will worry a little bit. Can you tell me what are the different tissues that they have to cross in order to reach their destination? For example, the oxygen. What does it cross? How many layers does it cross? It crosses this. What's this? The alveoli are made up of what? Is this a difficult question? The oxygen will have to cross the layers of the alveoli, and the alveoli, we know it is made up of a single layer of epithelial cells. So the oxygen will cross the epithelial cells lining the alveoli, these flat epithelial squamous cells of the alveoli. They will cross the walls of the capillary. You know very well that the capillary is made up of one cell thick. It's just one endothelium. They will cross the walls of the capillary, and then they will enter the red blood cell. They will cross the cell membrane to enter the red blood cell. Just the different layers, okay? Because this question is out of 3 marks. Write it down. Let's look at the answers to see what exactly do they want. Any 3 from diffusion. Okay, good. Movement down a concentration gradient. Good. In terms of, except in terms of partial pressure. We did mention that the oxygen is in the alveoli. In other words, it is highly concentrated in the alveoli. It's a passive process through the alveolar wall, across the alveolar cells, across the squamous epithelial cells. We said it. Across the endothelium of the capillary. We said it. Or the squamous epithelium cells. In context of capillary wall, we said the capillary wall referred to diffusion through the phospholipid bilayer of the cell membrane. I don't think we missed anything. Anyone has any questions? This question is more than easy. It can be an O-level question. Okay, if you have any questions, stop me. Okay, another graph. This graph also the partial pressure of oxygen and the percentage saturation of hemoglobin. This is another oxygen dissociation curve. This curve is so important. Okay, figure 4.1 shows the percentage saturation of hemoglobin changes at different partial pressures. Use figure 4.1 to calculate the difference in pressure saturation of hemoglobin at the lower partial pressure of oxygen of 2.7 kilo partial pressure compared to the higher partial pressure of 13 kilopascal. So, how will you calculate that? Show you're working. This is 1 mark. By the way, I don't want you to be confused. The difference in percentage saturation. Already, what is on the y-axis is the percentage saturation. And it's 1 mark. What will we do? I don't want silence. I want someone to give me an answer.
We
Where's
will make
Maryam?
final minus initial divided initial times 100.
No, we will not do that. We will only do final minus initial. Why? Because it is already a percentage saturation. Use figure 4.1 to calculate the difference in percentage saturation at the lower partial pressure of oxygen of 2.7 compared to the higher. So the lower is 2.7. My God. Okay, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So every small box is 0.2. So this is 2.2, 2.4, 2.6, 2.7 is nearly here. 2.7 is here. Okay, 21, 2, 3, 4, 5, 6. So 22, 24, 26, 28, 30, 32. Graphs in the AS are not as easy as in the O level. Okay, this is 2.7 and 13.0 kilopascal. Okay, 13, where's 13? 13, 12, 12.2, 12.4, 12.6, 12.8, 12 point. So this is 13. I'll go up, 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 up. Of course, I don't have a ruler here. This is 8.2, 8.4, 8.6, 8.8, 90, 92, 94, 94, nearly 94. So it's 94 or 96 minus 32. What's the answer? What's the answer? Who has a mobile next to him? Kenzie, you know why we did not divide and do all that?
Yes, because it's already a percentage.
Very good. Let me do it with you. 94
62.
minus
62.
62. Okay. Of course, I don't have a ruler, so I assume it's, let's look at the answers. It's 60 something. Okay. Yes. From 65 to 67. Of course, if I had a ruler, I would have gotten more accurate values, but we only subtract. We will not divide by 100 and divide by the starting end times 100. They want to confuse you. Explain the advantage of having a difference in percentage saturation of hemoglobin at lower and at a higher pressure of oxygen. Now, this question is so important. Why there is difference in affinity, and why is this an advantage? Who will try to solve this question? Do you understand the question? It's as if they are asking you, why, when we have little oxygen, the hemoglobin has low affinity to oxygen? And why, when we have too much oxygen, the hemoglobin has high affinity to oxygen? Why? Because this is what the oxygen dissociation curve tells you, that when I have little oxygen, the affinity is low. And when I have high oxygen, the affinity is higher. Yes, Mariam?
When the affinity is low, it is an advantage because the oxygen in the blood is less, so it then dissociates.
Excellent.
Yeah, it dissociates from the oxygen and therefore increases the oxygen level in the blood.
Very good. And it will release the oxygen and bind with what instead?
The hydrogen ions, preventing a decrease in pH.
Exactly. This question is very important. So please write it down with me. I will divide it into two parts. I'm going to talk about when it is of low affinity, when I have low levels of oxygen, and when I have high levels of oxygen. Exactly like what Maryam said. When I have low levels of oxygen, there will be low affinity. In other words, the hemoglobin will not bind with the oxygen. The hemoglobin will dissociate. The hemoglobin will break off from the oxygen. There will be dissociation. There will be high levels of dissociation. Why? Because this oxygen will be used where? What did you say, Maryam? This oxygen will be given to the what?
To the body cells or in the
Very
blood.
good, to the respiring tissues, so that this oxygen is used in aerobic respiration. And when the hemoglobin releases the oxygen, this hemoglobin will bind with what instead?
Hydrogen ions.
Very good. To give me hemoglobinic acid like Maryam said exactly, to prevent a decrease in blood pH. Now, Kenzie, tell me the exact opposite. When I have high levels of oxygen, what
I have
will the
high
hemoglobin?
affinity.
Excellent. I will have high affinity. High affinity means that the hemoglobin will bind with the oxygen to give me what?
Oxyhemoglobin.
Very good. And it will take this oxygen and give it to who? This oxygen will be delivered to who?
To the body cells.
Very good. The oxyhemoglobin will be delivered to the body cells so that they use it in, I'm writing with you, write with me.
Respiration and energy release.
Excellent. Delivered to the body cells for aerobic respiration. Now, of course, they are giving you a nice big graph. Please quote numbers from the graph. You can use examples. Example, when the partial pressure was 2, the percentage saturation was 20. The affinity was low. When the partial pressure was 12, of course, partial pressure is 12 at the lungs. Partial pressure as 2, this happens in the respiring tissues because the tissues are using up the oxygen and giving CO2. When the partial pressure is 12, the percentage saturation is nearly 90. You will definitely score the 3 marks. Okay, this question is nice and it's important because this is an explanation of the oxygen dissociation curve. Let's look at the answers. Okay, what's this? Where are we? Yeah, so let's read quickly, make sure everything is done. Higher partial pressure in the lungs, okay. Lower partial pressure in the tissues, good. Correct values, range of values. Yes, we took the values from the graph. Advantages of differences in higher vs lower partial pressure. Only higher pressure or lower partial pressure explanation required to gain mark. Oxygen binds to versus released from. This is what we said in detail when we made a table like this one here. Okay, we compared binding to and released versus released from. Oxyhemoglobin, we said it is formed, we said it versus dissociation, Maryam said it. So percentage saturation, no. Affinity of hemoglobin, we said it, data, we said data from the graph, body cells, tissues, they need the oxygen. Perfect. We actually gave a lovely answer. We gave a beautiful answer. We divided the two conditions and we gave numbers from the graph. Our answer can be a model answer. Anyone has any questions? Okay, anyone has any questions? Fine. In a person with sickle cell anemia, the ability of hemoglobin to transport oxygen and carbon dioxide is severely affected. The cause of this disease is a mutation in the gene coding for the beta globin polypeptide of hemoglobin. Define the term disease. Okay, we will take the definition exactly in the lesson diseases, but it's a very easy definition. A disease is anything that will make you unwell. It will change the conditions of the body, will make you not in your best shape. Okay, so you can write it. A disease is any abnormality. Are you writing with me? Or change in body processes so that it can lead to poor or ill health. This is one of the easiest lessons, by the way. You will have poor health, you will have signs and symptoms, and you can give any example. Example, lung cancer, God forbid, chronic bronchitis, anything. Anemia. Okay, very easy question. Now outline the differences between the hemoglobin A, the normal, and the sickle cell alleles for the gene coding for the beta globin polypeptide, and explain how these differences lead to a change in the hemoglobin molecule formed. Okay, this question, we studied this question in 3 lessons, by the way. We studied this question in biological molecules, in the lesson biological molecules. And then we also referred to it in the lesson circulation. And we will study it again in detail when we take mitosis and synthesis of proteins. So this objective is a very important objective. Look how many marks, 5 marks. What do you already know about this type of gene mutation? We did mention it in biological molecules and in the lesson transport in animals. Hello? It is in your notes.
Yes, doctor. Can we
What do
refer
you
to
or I can't hear you.
biological factors for the structure of
For the structure of the what?
the hemoglobin?
Yes, we did take it there. And then we did mention
It's
that
a
it is,
globular protein.
yeah,
It's formed of quaternary structure.
yes. Okay, maybe you forgot. We'll take it in the lesson protein synthesis and mitosis in detail again. Now, they want us to compare between the healthy hemoglobin and the sick hemoglobin and the sickle cell anemia. You already know the structure of the healthy hemoglobin very well. So let's also make a nice table here. Well, answer with me, okay? You have here the healthy hemoglobin and you
Can
have
you
the,
it's hydrophilic? It's a water soluble molecule.
yes, you can say everything. So this healthy molecule, like Mariam said, it's a quaternary protein. I'm writing down with you. Quaternary protein. In other words, it is made up of 4 polypeptide chains working together as one unit. And it has a heme group, and this heme group binds with oxygen so that it forms oxyhemoglobin. Okay, and bonded together by disulfide bonds, we have 2 alpha globin and 2 beta globin subunits. Now, this is the healthy. Sometimes there is a gene mutation. You have to mention mutation. This mutation, we will study different types of mutations, but we know that we have a difference in the normal base sequence. The normal base sequence is easy to remember. It is GAG, G-A-G, guanine, adenine, and guanine. This guanine and adenine and guanine, this is the normal base sequence. This normal base sequence results in the production of the protein. Who remembers the glutamic acid, amino acid? Okay, so the GAG or the base sequence GAG, because this is a base triplet. Every 3 bases code for 1 amino acid. Those 3 bases code for the amino acid glutamic acid. This glutamic acid is polar or hydrophilic, or it is water soluble. You can write anything you want. They all mean the same thing. In other words, like Maryam said, they have hydrophilic R groups, just like Maryam said. So they dissolve in water or they don't dissolve in water?
They dissolve in water.
Yes, the glutamic acid is soluble in water. Now we are talking about the healthy hemoglobin. Now we have a diseased hemoglobin. It's a genetic disease in which it will be caused by a mutation. We know that the normal base sequence is GAG, G-A-G, and this adenine will be replaced by the nitrogen base thiamine. So the mutated base sequence is GTG. Okay, I'm explaining it in detail because this is part of your syllabus. We will take it. And this question is important. Now, when this mutation happens, first you need to know it happens where? In the beta
globin.
globin. Yes, correct. In the beta polypeptide chain. Now, when this base sequence is mutated, we will no longer produce the amino acid glutamic acid. We will produce what instead? Another amino acid. Do you remember
Valine.
its name? Excellent, valine. You should have studied it for the exam. This could have been a question. This valine is completely opposite from glutamic acid. It's not hydrophilic, it's hydrophobic, and it is insoluble in water. And it has hydrophobic or non-polar R groups. Non-polar R groups. So this sickle cell hemoglobin, is it soluble or insoluble, Farouk? Hello? Farouk, where are you?
I'm here. It's
This sickle cell, soluble or insoluble?
It's soluble.
Insoluble. Okay, it is insoluble and it causes a lot of. This is, by the way, all this we took it. But when we study mitosis, we will take further information. We will see how this hemoglobin, sickle cell hemoglobin, affects the red blood cell. It will make it sickle in shape. It will make it sticky. It will make it block capillaries. It will make it carry less oxygen. Of course, less oxygen means poor ventilation, and the body will have increased rate and blood pressure, and this is causing a strain on the heart, and the same sequence that we already know. Okay, but this is not what they want. They want the differences between the healthy hemoglobin and the sickle cell hemoglobin. So we revised this part. Anyone has any questions? You need to know the normal base sequence and the mutated. This is called a base substitution mutation. We'll study the types of mutations later. The normal or the healthy base sequence is GAG. It's easy to remember, GAG, G-A-G. The adenine will be replaced by the thymine. Okay? Any questions? Okay, we finished half. Good. I want to continue quickly. Name the structures labeled A, B, and C. Farouk, what is A?
The trachea,
Yes, well done. What is B?
the bronchioles,
Yes. What is C?
the alveoli,
Yes. And this is so easy. Sometimes I worry when the questions are too easy. Name a non-infectious disease that affects the human gas exchange system. Now, we took
emphysema,
several diseases, and they are all non-infectious, by the way.
emphysema,
Emphysema, chronic bronchitis, lung cancer, cystic fibrosis, all these are non-infectious. They're not caused by pathogens. I think I showed you this question in class, and I told you that they like you to compare between A and B. Tell me, Kenzie, which one is a healthy individual? A or B?
A,
Yes,
A.
of course A. Why A? Why not B?
because the alveoli in A recoil back, and there are more of them.
Excuse me, my dear?
The alveoli in A recoil back all the way, and there are more of them.
There are more of them, more alveoli. And when you have more alveoli, and obviously they're smaller, they're not large and dilated like in B, how will this affect the surface area?
It increases the surface area.
Yes, more surface area to volume ratio, better gas exchange. Okay, now B is the opposite. They're larger because we have air trapped here, and they have less surface area to volume ratio, less in number, less capillaries. Of course, this happens because the elastic fibers have become destroyed because of the elastase enzyme and the story that we already know. Now let's see what they want actually. Name the structure labeled H. Now this is not what they want anyway. What's H? What is H? This flower. I told you the tissue or the structure that looks like a flower is the what? I showed it to you in the PowerPoint. It has no cartilage. Yes, very good. It's the bronchiole. Not bronchi, bronchiole. The bronchiole, remember, it looks like a flower. You have lots of pictures. You have to know the histology pictures. Calculate the actual diameter of the alveolus XY. Write down the formula and use it to make your calculation. Give your answer to the nearest whole micrometer. Okay, they have a box here for the formula. What's the formula?
Image
What's the
over
formula?
magnification.
Yes, I am. What do they want? They want the actual. So it's the image over the magnification. What is the magnification? The magnification must be provided. Let's look at the question. Here, the magnification is times 40. So the magnification is times 40. I already know that. Now, the image, how will you do it, Kenzie? How will you get the size of the image?
We will calculate it in centimeter, then convert it times 10 to millimeter, and
Correct.
then convert the millimeter to a micrometer by
Excellent.
multiplying
Very
the
good.
value by 1000.
Very good. I will measure it in centimeters times 10 to millimeters. Even this is O level times 1000 converted to micrometers. Of course, our answer will not be the same as the mark scheme because the dimensions will change. Gas exchange in the lungs occur in the alveoli. Describe the process of gas exchange in healthy lungs and suggest how this compares with gas exchange in the diseased lung shown in figure 1.1b. How many marks? 4 marks. Now, this question is so easy, but sometimes you don't understand what do these people want. When they ask you describe the process of gas exchange, remember the question that we answered and we said the direction or the movement of the gases. We said oxygen moves from the alveoli to the capillaries by diffusion, and the CO2 will move in the opposite direction. Remember when we said that? This is the same answer, by the way. You will mention the direction of motion of the gases and how do they move by diffusion and the pathway. What do they cross? The squamous epithelium of the alveoli, the capillary wall, the red blood cell. But here, they want you to compare. Describe the process of gas exchange in healthy lungs and suggest how this compares with gas exchange in the diseased lungs. So we will describe what happens in the healthy lungs. And we will compare that with diseased lungs or diseased alveoli. Now, we have to mention here a particular tissue that will help or cause the alveoli to become wide like this. What happened to make the alveoli become wide and not be able to expel the air and decrease in number? What's the reason the alveoli changed from A to B? Tell me.
The elastic fibers are broken down.
Exactly. You will mention the elastic fibers and their function and what do they do in normal situations and what happened to them in B to make all this change. Okay, so you will mention the normal pathway and you will mention how will this differ when the elastic fibers are destroyed. All this will decrease. We said that in A, we have lots of surface area to volume ratio, everything is nice, and the oxygen will diffuse down a concentration gradient, and the CO2 will diffuse in the opposite direction, and there will be efficient gas exchange, and we will have good ventilation. When you are diseased, the first thing you will mention that will be affected, the elastic fibers, just like Mariam said, elastic fibers will become destroyed. So when they become destroyed, what happens?
The alveoli cannot stretch and recoil.
Very good.
When the air enters, when we are inhaling and the air enters the alveoli, it overstretches and it cannot push out the air during recoiling because there are no elastic fibers. Therefore, it bursts.
Excellent, excellent. And you will say, of course, this will have consequences. This will decrease the surface area to volume ratio, increase blood pressure in the lungs. You can say so many things. It will affect, if this remains, this will put strain on the heart, causing the right side of the heart to enlarge. If this also is not treated and it lasts for a long time, maybe for years, this can cause coronary heart disease. This question here, you can write like a whole page. Now let's look at the answers. But do you understand what you will do? You will compare healthy with the diseased, and you will explain why and the consequences. Let's look at the answer, and I want you to maybe not write the whole thing now, but write the main points. Where is this question? Here. No. Where? This one. Okay, see all this. Let's see what do these people want. Okay, any 4 from, this is out of 4 marks or 5 marks? 4 marks. In healthy lungs, the correct direction of. Okay, I don't know what happened to the connection. Are you all with me?
Yes.
Okay. Now, let's look at this answer. We will compare, we said the healthy with the diseased. Okay, correct direction of movement, we said it. Example, oxygen moves from alveolus towards it, we said. Excuse me? It's not shared?
Yes.
Oh, okay. Thank you for telling me. Okay, this actually is an easy question, by the way. Or movement down a concentration gradient from high to low, implied oxygen diffusion. We said all this and the detail of the pathway, reference across the alveolar wall. Yes, this is exactly the same answer as the last question. Do you realize that? Exactly the same answer. How the gases move, in what direction, and what do they cross. But in addition to that, we will mention What happens when we're sick? Where is the part where accept squamous cells comparison? Healthy with diseased. Higher rate of exchange, increased rate of diffusion, we have better oxygenation, larger surface area. We said all this, shorter diffusion distance, okay. Reference to greater ability to stretch and recoil, very good, because we are healthy. Now, all this in healthy lungs. Correct direction of movement of blood, we said it. Oxygen from the alveolus towards, we said. Or movement down a concentration gradient from higher to zone. We said all this. Why are they repeating themselves? Actually, maybe this is my mistake. I'm the one who wrote the answer more than once. Higher rate of exchange, okay. Now, do you have any questions? This answer is just repeated more than once. It has been copied more than once. Any question? Very easy question. You will mention the direction of movement, you will mention diffusion, down concentration gradient across the different layers, and we even added the functions of the elastic fibers that will be destroyed. Any questions? We're very slow. Any questions?
No.
Okay, let's answer this exam. Some tissues of the gas exchange system include cells that are able to produce and secrete mucins. Mucins are stored in vesicles in these cells, ready for secretion. Once outside the cell, mucins absorb water to form mucus. Okay, now this is a tricky question. Here they gave you a new concept. This concept, we do not study it in the AS, we study it in the A2. Those that take A2 in the lesson homeostasis, they study this concept. Now let's see. I want you to focus on this sentence. Mucins are stored in vesicles in those cells, ready for secretion. Once outside the cell, mucins absorb water to form mucus. So, in other words, you have cells that will release this gluey material or the mucin. And then water will follow. Why is it important that water follows?
For
What do you
solvent,
think? Excuse me?
water acts as a solvent.
Okay, you are correct, of course. Water is an international solvent, and the water will dissolve the mucin a little bit and make the mucin thick or thin.
Thin.
Yes, correct. It will make it thin. It will dilute the mucin a little bit, converting it into mucus. And why is this important? So that who can sweep it away? Who will sweep the mucus away, Mariam?
Yeah.
The cilia. Remember the disease cystic fibrosis when we said that we have very, very thick mucus that cannot be swept away by the cilia and it accumulates and it causes a lot of problems? Remember?
Yes.
Very good, good. Put it in mind. A, name the structures and the gas exchange system that produce and secrete mucins. Come on,
Goblet
what's the name? Goblet cell, of course, or mucus glands, goblet cells. I don't know why this is 2 marks. Okay, goblet cells, 2 marks, then you have to say where is it found. You have to add another fact. Goblet cells or the mucus glands, because it's 2 marks. Mucus glands, it's found in the trachea, bronchi, not in the bronchioles. It's definitely 2 marks for a reason. Okay, mucins are described as the glycosylated proteins that contain carbohydrates. The process of glycosylation, again, we take glycosylation in A2, involves the addition of sugar components after polypeptides are synthesized. Suggest one location of the cell where glycosylation of mucin would occur. Tell me, Kenzie, where does the modification of proteins take place? When proteins are made, they are modified. They are modified where?
I think the Golgi body.
Yes, in the rough endoplasmic reticulum or the Golgi body. Okay. The processes that occur in the production and secretion of mucins are listed. You have translation, exocytosis, glycosylation, and transcription. We still did not take protein synthesis, but I think we took it last year. We were introduced, we said that the DNA is copied on a messenger RNA. This is transcription. And then the messenger RNA will go out of the nucleus and bind with what? Who remembers? What is the organelle that will do the translation? What is the organelle that produces proteins?
The
Ribosome.
ribosomes.
Yes, the ribosomes. The ribosomes, we will have the translation. Complete table 2.1 by writing the processes in the correct order by which they would take place. Now, what's the first process? I want to synthesize a protein. We still did not.
Translation.
No, translation is not the first process. First, I will take a copy of the DNA. So the first process will be transcription. Transcription, this is when I will take a copy of the DNA. And then, after I take a copy of the DNA, I'm going to go to the ribosome and translate it. So this will be the second process, translation. Write it down with me. Okay? And then the polypeptide will be made. I don't expect you to know this now because we still did not take protein synthesis. And then modification of the protein will be made, which is glycosylation. Glycosylation. And the fourth process will be its release outside the cell, which is exocytosis. Okay? We still did not take protein synthesis, but this is the correct answer. Transcription, translation, glycosylation, exocytosis. Let's see what do they want next. Chloride ions move. Let's solve this question. It's a very important question. It's a suggest question. Chloride ions move out of the mucin producing cells at the same time as mucin is secreted. Suggest and explain how the exit of chloride ions helps the formation of mucus from mucin. 3 marks. Now I want you to understand this is a new concept that they are introducing to you. This is a suggest question. Once you see the word suggest, know that you did not take it. But this is a critical thinking question. So they're telling you that I have a cell. This cell will release mucin. And then chloride ions will move out of this mucin and then it will release chloride. Now why?
It makes it polar, therefore it attracts water more easily.
Okay. Part of your answer is wrong and part of your answer is correct. It attracts water, yes. But what's the relation about what's polar? What's polar? This is the inside of the cell and this is the outside of the cell. When I have more chloride ions outside the cell, it will attract water, correct. But why? Water moves by what?
Cohesion.
By what?
Osmosis.
Of course, I have water here. Water will follow the movement of the chloride ions by osmosis. Now, why does the water follow?
Because the water potential inside the cell is more than outside the cell.
Correct. Very good. Because the water will move from high concentration from the inside of the cell to low concentration, which is outside of the cell. And this is a very important step because this will dilute the mucin and change it into lighter mucus. And when the mucus is light, now it can be swept away by the cilia. Because if it is not swept away, mucus will accumulate. And we will go back to the story that we mentioned in class. It will accumulate, causing infection, attracting macrophages that will release what? Elastase enzyme that will try to create a pathway to reach the site of infection. But while doing so, they will break down what, Kenzie? Where are you? They will break down elastic fibers.
No, elastic fibers.
Very good. Now you know this part. But this question is critical thinking. I am worried you will not be able to answer that question. Do you understand it, girls? We have a goblet cell.
Yes.
Okay. If you will take A2, you will know that this is a two-step procedure. The goblet cell will release mucin. Mucin is very, very thick. The mucin will attract the bacteria. We don't want the mucin to remain there. We want it to go away. But we have to dilute it first before it goes away, before the cilia sweeps it away. So the goblet cell will secrete chloride ions to make the concentration outside the cell higher than the concentration inside the cell. Like Mariam said, water potential inside the cell will be high. So this will make the water to move outside the cell to follow by osmosis and dilute the mucin. And when it is nice and thin, it will be swept away by the cilia and it will not accumulate and cause infection. What do you think? Clear?
Yes.
Very good. Okay, let's finish this question. Go to the mark scheme later. The gas exchange system includes some cells that are able to divide by mitosis. Explain why it's important to have these cells in the gas exchange system. Again, we did not take mitosis, but we can still answer these questions. Some cells are found in actually every organ in your body. These cells, they can divide by mitosis, and they're very important. What do they do? What are these cells anyway? If you think of your skin cells, these skin cells that are always falling off, how are they replaced by new skin cells? Who would replace them?
The unspecialized cells.
Perfect. Perfect. In every organ in your body, you have unspecialized stem cells. These unspecialized stem cells are of the adult type. We will study stem cells again. But I think I mentioned adult stem cells. These are stem cells that specialize to a particular type of cell. They already went through some stage of differentiation, but they have the last stage. They differentiate into a genetically identical cell. You have cells that have been damaged. They have to be replaced. Who will replace them? Those adult stem cells. They divide by mitosis in order to produce more stem cells or specialize cells that are genetically identical to the cells that have been damaged or lost. So these cells are very important because they replace damaged cells or they repair tissues. And they are present in some organs more than the other, like the skin. The skin has a very high regenerative power. When you cut your skin, a few days and the skin will repair itself. But some tissues or some organs don't have them, like the nervous system. And unfortunately, if you destroy your nerve cells, God forbid, you don't have stem cells that will differentiate into new nerve cells. That's why people that are paralyzed, they don't have a cure. Okay, but we do have some of them in the gas exchange system, and they divide by mitosis to replace the damaged or the dead cells, and we have to replace them with genetically identical cells. Okay, and of course, they're needed to replace damaged cells or to repair tissues. They're needed for growth. They're needed to just repair any worn out cells. Okay, we will take them in mitosis, but you already know a question like this. You can actually answer it. Let's look at the mark scheme. Where are we? Oh, this is the question. To produce new daughter cells that are genetically identical, to replace all damaged or dead cells, to repair tissues, any tissue. If marking MP2 is marking 0.2 and 3 not gained, allow 1 mark for replacing damaged tissue, reference to function, any valid point. You have to mention the idea of increase in the number of cells or the growth of cells to accommodate increase in size of the gas exchange system in growth, infants, alternative wording, any one of those. So I think Kenzie, she gave us the answer. She gave us the beginning. Okay, do we continue? Or you can solve the rest on your own? 20 out of 30. You have 10 pages. So solve the 10 pages on your own. We solved 20 pages. That's very good, or 19. You have nearly 11 or 12 pages. Solve them for homework, please. Please. And if you have any questions, ask. Okay? This is one of the easiest lessons. The questions are very easy. The questions repeat themselves. Okay. Okay, excuse me, my dog is breaking the walls down. 1 minute. Okay, I'm back. Now we have Filfil joining us now. Okay, we have 10 pages left. Please solve them for homework, and if you have any questions, ask me. Okay?
Okay.
Okay, Kenzie, you have any questions?
No, thank you, Doctor.
Welcome. Bye. You want to see Filfil? خلاص. Girls,
Yes, doctor.
you want to see Filfil? He wants to say hi. No, he left. خلاص. Bye bye.
Bye.
Bye.